right, we are recording. Let's see here. Kip, David, Hodges, Sipple, Eric. Midkiff is here. A little extra credit for physically being present. Same with Rice. Same with Massey. Present. Felder's online. Tudor's present. Physically present. So was Asbury. So was Michaela. So was Kinsey. Is it Hemmings? Nope. Hold on. Let me guess. It makes it hard. The online option makes it where I don't know everybody's name. I apologize. Not Richardson. Hold on. I'll get this. It's a multiple choice question. Bancross. No? All right. I give it. That's right. I guess I would have gotten there next. Physically present. Burdette is physically present. A little extra credit for that. Richardson? Got one right. Physically present. Got a little bit more than 2 minutes left. Does anybody have any announcements before we get started? Anything you want to share with the class? All right, makes things easier. I'll make some announcements for attendance. Obviously, I haven't posted the video from Friday yet, so I guess that's my fault. If you haven't, turn it in like right. If you don't have attendance points for Friday, you can watch that and take care of that. What else? Today will just be like regular attendance. Any questions about that? All right. The other one, just reminders. So for lab, don't forget your pre-lab has the questions and then the 2 videos. So make sure you do those. Technically, it's past due. That was due last night. But as I always say, as long as it's done before I get around to grading it, I consider that on time. And don't forget, if you do the whole lab, at the very least, you'll get extra credit. And there's a possibility you might not have to come to lab this week. Some of you have already done that and know that you don't have to come to lab this week. So go ahead and do it if you want. Let me know if you need help. And also, because it's an online lab that we're meeting for in person, make sure to bring your computer to lab if you have one. If not, I'll provide you with one. It's going to be a crappy old one, but I'll give you something. Yeah, and that's it. Also, it doesn't matter what day you come. You come on Monday. I had to get. Let me know. Show up on Monday. Show up on Wednesday, or like I said, if you do it beforehand, and I grade it, I might tell you you don't need to come in at all. So, any questions about lab? All right. I see Sipple and Van Cross have joined us online. Sipple, Van Cross. Anyway, what other reminders? Since we still have a week or a minute, Cedric is online. I'll remind you that the exam is on Wednesday. It's online, so we're not going to be here in person. Don't come to class on Wednesday. unless you want to, in which case let me know beforehand. Otherwise, I'm going to be in the office instead of in here. So you can take a paper version of the exam if you want. And again, it's 50 minutes, right? You have from 8 to 8:50, just like you normally would if we were meeting in person. So if you want more time to take the exam, let me know sometime between now and then, so we can arrange a time to meet online, so I can make sure that you're taking closed book exam. As long as I know you're not looking at the internet or your notes or your books or whatever, I don't care. Exam review. I was watching that video that I've already posted, and I'm going to change things up. We're not going to, but this is actually more work for me. Once you've done the study guides, if you have questions, I will sit down and do an exam review with just you. Like if you say, all right, I've done the study guides, can we meet and go over these questions? Then I will do that. So let me know. Any questions about that? All right, I think that's all the reminders. Lab, exam, independent work, go ahead and get it done. Points per page. It's currently worth 16 points per page. It's going to start going down again soon. Hodges is online. Bowers is online. Any questions about anything? You guys are making it easy. All right. So again, one last time, the exam is on Wednesday. It'll be online, 8 a.m. Make sure you're taking it. Make sure you submit it by 8:50. Make sure you don't miss it. Newton is online. The exam's going to cover chapters 5, 6, and 7. It's now 8:01, so we will get started. I guess the first word for attendance will be exam, since we're talking about it. Exam is the first word for attendance. Let's jump back into chapter 8. And to remind you, this chapter is not going to be on this upcoming exam, right? This is not going to be on exam number 2, the one we're going to have on Wednesday. We're kind of skipping ahead here. I'll also remind you what we've talked about. So again, we're talking basically about cellular reproduction. So if you remember way back in chapter 1, we talked about the properties of life. One of those properties of life was that life reproduces. So that's what we're talking about, right? Cellular reproduction. We talked about what it accomplishes. So in that first main bullet point, you kind of learned the difference between sexual versus asexual reproduction. You learned about the basic difference between mitosis and meiosis, but we're going to get into more details with that moving forward. Then we got into the second main bullet point, which actually gets into the cell cycle and mitosis. And technically, we haven't even covered the cell cycle or mitosis yet. We talked about eukaryotic chromosomes. Talking about like humans or plants or fungi, right? The complicated life, not like bacteria, right? So there's prokaryotic, there's eukaryotic. We are eukaryotes. So we talked about eukaryotic chromosomes. Then we talked about duplicating chromosomes, and we're going to get into more detail with that too. But to remind you, chromosomes obviously have to duplicate before the cell splits. Otherwise, the cell is going to have half the number of chromosomes, and that wouldn't work, especially generation after generation, right? So happens. Actually, we'll get into when that happens here in a second. And that's where we left off. So now we're finally getting into the cell cycle, and then finally we'll talk about mitosis. And then after that, meiosis. So any questions about where we're at before we jump into it? All right, let's talk about the cell cycle. It is the cell is first formed until it splits again. And in a sense, you can basically think of it as the life cycle of a cell. So unlike humans or any other animals, the life cycle is when you're born versus when you die. It's not like that, right? Because with cells, it's where it starts, and then when it splits, that's the life cycle. So that's the first thing you need to know about the cell cycle, right? From the moment it begins. Questions about that? Pretty simple, right? Here's what it looks like. And then we have S phase, then we have G2, then we have the mitotic phase. So that's the first thing you need to know about the cell cycle, is know the different phases and know what order they are in. And soon I'm going to teach you what happens in some of these phases. Mostly what I'm concerned about is what happens in the S phase, which you're going to learn here in a second. You can see up there, that's when the chromosomes duplicate. That's basically all you need to know about it. And then I'm going to teach you, and you need to know about the mitotic phase. The book also tells you what happens in G1 and G2, so therefore I will too, but I'm not going to test you about it. So a common mistake I see when people talk about, or this part, they'll talk about, you have to put it in order, right? There'll be questions like, I put the stages of mitosis in order. And I don't think I'm going to do this, but just in case I do, if interphase is an option of the multiple choice questions, don't use interphase. Because yes, interphase does come before the mitotic phase, but it also comes after. Technically, interphase is not part of the mitotic phase, right? So the whole cell cycle, you could basically say it's broken down into two parts, interphase and the mitotic phase, right? So all these three things, G1, S phase, G2, that's all considered interphase. So again, when you're studying, that's what you need to know. First of all, like I usually recommend, study the big picture, then start adding details, right? So the whole cell cycle is broken down into interphase and the mitotic phase, right? And then once you have that memorized, then memorize the fact that the interphase itself is broken down into G1, S, and G2. And then, of course, you're going to learn that mitotic phase is also broken down into different stages. Also, I'm not even going to ask you this. Yes, technically, cells spend more of their time in interphase than they do the mitotic phase, and I'm not even going to ask you that. Details on it. All right, let's do it then. Like I said, the cell is mostly in interphase, right? So the cell's usually in interphase. If you were to look at all your cells, somehow if you could do that, most of the ones you would see would happen to be in interphase. Some of them would be in the mitotic phase. Those of you who've already done the lab for this week, you've seen that, right? You've seen all those pictures of cells, and that was an accurate representation because it kind of gave you a graph at the end that you had to take a screenshot, and you could see that most of them were in interphase. So what happens during interphase? This is basically when the cell just does whatever it does. So if it's a kidney cell, it's doing kidney cell things. For brain cell, it's doing brain cell things. Whatever, right? That's just doing its normal thing. And as a matter of fact, I'm not even going to ask you what happens while it is in interphase. It's good to know, but I'm not going to ask it. Just doing its normal cell thing. Of course, the mitotic phase is when it splits, right? And that's mostly what we're going to cover in the exam. And obviously, before it splits, it needs to get ready to split. So yes, obviously, it's going to grow in size, right? It's going to double everything. Double the mitochondria. If it's a plant, it's going to. But again, for the exam, I'm not going to ask you what happened during interphase, except for S phase, and we'll get there here in a second. But overall, just know that interphase is the other part of the cell cycle other than the mitotic phase. Any questions about that? Pretty simple stuff, right? S phase, again, the whole cell cycle is broken down into interphase and the mitotic phase. And if we're S phase and G2. And S phase is the one we're more interested in because a lot of stuff happens when cells divide, but what we're mostly focused on is the chromosomes, right? So when we talk about mitosis, that's mostly what we're going to talk about. What happens to the chromosome? Right? We're focused on the chromosomes. Therefore, that's why we are so focused on S phase. Because S phase is when the chromosomes duplicate. So that's part of the chromosome story. And that's mostly what we're into. Speaking of which, for independent work, you might look up what else happens, right? So again, we're focusing on the chromosomes for mitosis and the cell cycle. But what about the rest of it, right? What else is happening during the cell cycle? We're only telling the story of the chromosomes mostly. But anyway, again, for the exam, for this slide, what you need to know is when do the chromosomes duplicate? S phase of interphase is the answer. S stands for DNA. And again, maybe you're going to need to know the order. So you might need to know that S phase comes between G1 and G2. I don't know if I'm going to ask that or not yet, but definitely know that S phase is when the chromosomes duplicate. So any questions about this slide? You guys must be tired. I'm tired. All right. So again, G1, G2, your book teaches about it, so I will, or teaches it, so I will too. I teach out of the book. I'm not going to ask you any questions about it, though. Let's get into it really quickly. The G stands for gap, so that should make it easy because, again, we're focused on the chromosomes, right? So we're focused on the mitotic phase, which I haven't taught you yet, and S phase, right? These two phases here. So what we're talking about right now is the gap in between them, right? The first gap and the second gap, G1, G2. I'm not going to ask you any questions about it, but there you go. Now you know what order they're in. G1's really when your cells are just doing the normal cell thing, whatever kind of cell they are. They're just chugging away. Again, like if it's a kidney cell, it's just doing kidney cell things. Then obviously if it's S phase, it duplicates its chromosomes. G2, it's really getting ready to duplicate the entire cell. But again, I'm not going to get into that. You can do it for independent. So you can look into it if you want. What other things happen in G1 and G2? Or, and I'm going to hint at this later, but I'm not going to give you the complicated information, but what controls this? Does it just go? Is it like on autopilot? Do cells just naturally go from G1 to S to G2 to metaphase, good grief, mitosis? Or is there something controlling it? All right, here we go. The mitotic phase. This is where it gets a little bit more confusing. Not much, but a little bit. And again, I want to take a step back and make sure you understand this thing in context. So of the whole cell cycle is broken down to interphase and the mitotic phase, right? So we just finished talking about all the different parts of interphase. Now we're talking about the mitotic phase, right? One of the two major parts of the cell cycle. And the mitotic phase itself is broken down into two processes. And they overlap. So towards the end of mitosis, cytokinesis is starting. And there's no reason, I don't think, to write any of this down yet, because we're going to get into a lot of detail about mitosis and cytokinesis. This is just an introduction. As you already know from the previous lecture, if we're talking about mitosis, this gives us two genetically identical daughter cells. Unlike meiosis, which I'm going to teach you later, because meiosis gives you 4 genetically unique daughter cells. So the numbers are different, and the fact that they're genetically unique is different for meiosis. Mitosis is all, or at least we're going to focus on what happens with the chromosomes, and then cytokinesis is when the cell itself splits. And as I'm going into this, I'll go ahead and let you know there's going to be 2 different questions, and people usually miss this. I don't do it on purpose to trick you, so I'm letting you know now. One of the questions is going to say, all right, put all the steps of mitosis in order, right? So as you're about to learn, it's prophase, metaphase, anaphase, telophase, right? That's it in order. And then there's going to be another question that's where it's out of order, and it says, in this phase, the chromosomes do this. In that phase, the chromosomes do this. In this phase, the chromosomes do that. And you're going to need to be able to pick out which of the 2 phases it is based on the description of what the chromosomes are doing. And inevitably, I would say historically, maybe 40 to 50% of you are going to just put it in order: prophase, metaphase, anaphase, telophase. So I'm telling you now, 2 different questions. One of them is asking you to put them in order. The other one's asking you to define them or recognize them by what's happening to the chromosomes. So any questions about this slide before we actually jump into it? All right. The next word for attendance, let's see, will be this. I'm going to circle it. I'm not even going to say it. So if you're watching the live stream, make sure you open your eyes and look right there, because that's the next word. If you're watching the video that I'm going to record and hopefully post later today, and you're not live streaming it right now, then take a screenshot of me pointing to the word "to." There we go. All right. Moving forward. let's talk about mitosis and cytokinesis. Again, like I said, you're going to need to know them in order. So the first thing you should memorize is this: prophase, metaphase, anaphase, telophase, or telophase, depending on who you talk to. Those are the orders of mitosis. So again, don't get it confused. Yes, interphase does come before prophase, right? Because that's a part of the cell cycle, but it's not a part of mitosis, right? It's a whole distinct part. Interphase is literally the opposite of mitosis, right? That's the part, the only other part other than the mitotic phase. So don't confuse that. Yes, also comes afterwards too, right? But it's not technically part of mitosis, so don't mess that up. So again, the story I'm going to tell you is all about the chromosomes. You need to know what happens with the chromosomes. And for independent work, you can look up and find out what else is happening, because this is pretty much all I'm telling you. Is like the chromosomes do this, the chromosomes do that. But what about the rest of the cells? What about the organelles? Any questions yet? Oops. Oh. All right. Let's move forward. We'll actually get into these. First thing I'm going to tell you before we actually talk about prophase, metaphase, anaphase, and telophase is I got to teach you about this thing called the mitotic spindle. I'm not necessarily going to ask questions about the mitotic spindle. I'm not sure, so I'll put a question mark next to it. But I am going to be describing prophase, metaphase, anaphase, and telophase. And in that description, sometimes I'm going to discuss the mitotic spindle. So for that reason, you need to know what it is. And again, I might ask about it on the exam, but I'm not sure. I'm definitely not going to ask you what it looks like. So yes, your book does say it's a football-shaped structure of microtubules, but I'm not going to ask you that. Separation of the daughter chromosomes. In other words, like I keep saying, for mitosis, for the exam, I'm going to keep asking you, the chromosomes do what, right? In this stage, the chromosomes do this. In this stage, they do that. So basically, they're moving around. That's what this whole story is about, right? Where the chromosomes move around. And it would not happen without this thing called the mitotic spindle. Mitotic spindle is just a thing, or the stuff, if you want to put it that way, that's moving the chromosomes around. So this is a picture of it. These right here are the chromosomes that I'm circling. And then you can see they have little lines attached to them, right? Little ropes, little wires, however you want to describe it. And those things attach, and those are the things that move the chromosomes around. But that's what a mitotic spindle is. Any questions about the mitotic spindle? All right. Let me ask you this. Imagine this was right here on this desk instead of on the screen, right? So imagine I had a bunch of, well, let's just start with this. Maybe two tables. We got this is a bunch of balls of yarn right here in the middle. And then I asked Kinsey here to separate it. I'm like, all right, you got all these balls of yarn. I need half of them on this side of the table, half of them on this side of the table. Meanwhile, assume on this table, actually assume the table's the same size. You have this, right? The same amount of yarn, but you have this squiggly mess. And I've got it right there in the middle of the table. And I say, someone else come here and split that. That makes sense? Like I'm asking both of these people to split the yarn and have half of it on one side of the table and half on the other. Who's going to win that race? The person doing this, A, or B? Who do you think is going to be able to separate that yarn the quickest? A, right? Because it's all nice and neat. It's packaged and this is little balls. That'd be really quick and easy. This is a squiggly, tangled mess, right? So keep that in mind when we talk about prophase. Prophase is the first phase that you need to learn about. It's the first phase of mitosis. In prophase, one of the things that happens is the chromosomes coil up, right? Because they're about to be separated. So you don't want a big squiggly, yarny mess, right? You want little balls of yarn, so to speak. And that's what happens. Logically, that should make sense, right? Logically, you should be able to memorize that. That should be the first step. Because before you can separate them, they need to be easier to separate, so they coil up. As you already know, because we talked about this when we talked about the S phase, the chromosomes have duplicated already, so technically they're sister chromatids. They're joined at the hip, so to speak. The proper term for that is the centromere. Imagine we're doing this, have this. All right, here we go. I'm going to pick on Kinsey again because she's here. Imagine I had a Kroger bag with this yarn in it, right? So sort of like what I said before, except now I've said, now it's in a Kroger bag. Right, so I got that bag or those things in the Kroger bag right here. And I say, all right, can you please split it? What's the first thing she's going to do to split this yarn? Again, she's got a Kroger bag full of balls of yarn. What's the first thing she's going to do? Open the bag, right? Or get it out of the bag, right? She can't separate it if it's in the bag. So logically, this would make sense too. This is also what happens for prophase. The nuclear envelope, the thing that makes up the nucleus that keeps those chromosomes inside, it basically disappears as far as we're concerned, right? It breaks into pieces. So now those chromosomes are able to be separated because, spoiler alert, the whole purpose here is they're going to go to separate sides of the cell. And to do that, they need to be outside of the nucleus. Therefore, the nuclear envelope basically disappears. Of course, for independent work, you could look up, where exactly does the nuclear envelope go? Of course, for this conversation, it doesn't matter. Just know that it basically disappears in prophase. Also, like I keep saying, mitosis is all about moving around those chromosomes, right? And you're going to need, that's how the questions are going to go. Like, in which stage are they chromosomes doing this? In which stage are the chromosomes doing that? So if the chromosomes are moving around, then obviously the thing that moves them around needs to attach to it. So that's the other thing that happens in prophase. The spindle attaches to the centromeres. As a matter of fact, step, the spindle is attaching itself, or itself, if you want to say that, to the chromosomes because they're about to move the chromosomes. So this is all very logical, right? This should make sense. This all has to happen first. The chromosomes have to coil if we're going to separate them. The nuclear envelope has to disappear if we're going to separate them. The spindle has to attach if we're going to move them. Now, the one part that's not as intuitive as the rest is this last bullet point, which is that the chromosomes are, at this point, after the spindle's attached, after the nuclear envelopes disappeared, so prophase and telophase. The first phase and the last phase are probably the hardest to remember, only because they have so much going on. The rest of them, as you're going to see, is very simple. So again, chromosomes coil up. That's logical. They need to. Nuclear envelope disappears. Again, very logical, very intuitive. The spindles attach, obviously, because that's the thing that's going to move them. And then I guess you might want to say the most important part, maybe, is the fact that the chromosomes are now moving towards the center of the cell. Now, let's see who's our thinkers here. If prophase is defined, if you will, by the chromosomes moving towards the center of the cell, right? That's where they're doing. They're going towards the center of the cell. What do you think the next phase is going to be? Where do you think the chromosomes are going to be in the next phase? So after this, where do you think they're going to be? In the center of the cell. I think you said that a little bit loud, right? So in prophase, if they're moving towards the center of the cell, then the next phase, they made it, right? By definition, that's just them. They made it. I'll show you a picture, or we'll talk about it here in a second. First, here's a picture of prophase. Like I said, the interphase comes before that. That's not part of the mitotic phase. That's the other part of the cell cycle, but that's what it looks like, right? So we've got this squiggly mess of chromosomes. We have the centrosomes, which I haven't even taught you about. That's where the centromeres, the spindles come from, excuse me. Then we get into prophase. Again, like I said, the chromosomes have to coil up because we're going to separate them. The mitotic spindle starts forming because it's going to need to attach, and eventually it does attach, right? The mitotic spindle is attached. The nuclear envelope has disappeared. Again, the chromosomes are coiled up. And what you cannot see in this picture is they're all, maybe you can't see it, they're all kind of moving towards the center of the cell. Of course, it's a picture, not an animation, so it might be hard to see that, but that's what's happening, right? They're moving towards the center of the cell. And like someone already said, the next phase is this. They made it, right? So in prophase, they're moving towards the center of the cell. And then once they get there, we call that metaphase. And the way I always remember that as a student is like that's where they meet. Right? They met up in the middle of the cell. Metaphase is when they met up in the middle. Of course, that's not what it means. That's a Greek prefix, meta. You can look it up for independent work if you want. But for the purposes of this discussion and for the exam, you could just think of it as that's where they met in the middle. And basically, for the exam, it's that simple. That's all I'm going to ask you. In what stage do the chromosomes line up in the middle or meet in the middle? I don't know how I'm going to word it, but if the question is, when are they in the middle? The answer is metaphase. That's simple, right? Now, what you cannot see in this picture, yes, they're in the middle, but what you can't see is these spindles are pulling it, right? So it's like a tug of war going on right here. They are pulling those sister chromatids. So if metaphase, well, first of all, again, it's defined by the fact that's when they got to the middle, right? And now I'm telling you they're getting pulled. Those chromosomes are getting pulled in opposite directions, those sister chromatids. So what do you think will happen in the next phase? If right now they're getting pulled in opposite directions, what do you think the next phase, what do you think is going to happen to the chromosomes in the next phase? No guesses? What is it? Opposite side, yes. Specifically, there's a picture of metaphase. Specifically, those, because of there's that tug of war, those sister chromatids are finally split apart, right? Because remember, they duplicated. So this used to be attached, right? Back in metaphase, that's chromosome, we'll call it chromosome number 1. And those were attached, right? Those were sister chromatids. And they were pulling at each other in the last phase, right? In metaphase. Now, finally, they split apart, which makes sense, right? Logically, that makes sense. There was a tug of war happening. They were getting pulled in opposite directions. And then once they finally break, that makes it anaphase, the next phase. The stupid way to remember this is you could say, like, think of it as like a relationship, right? So a couple met. First they met. Then some girl named Anna came into the picture and they split up. They started going and that's it. They split. Something you could look up for independent work is, does this happen all at once? Is it like if you were to look, if you could watch a cell in metaphase, would you see that they just bloop, all break up at once? Or is it like one chromosome and then another one? How does that work? And if it's not all at once, then when do you consider it anaphase, right? Is it when they're all separated? Or is it when the first one starts to separate? You can look into that for independent work if you want. For the purposes of this 100 level non-biology major class, you don't need to know that. All you need to know is once they've separated, we call that anaphase. All right. So what you cannot see in this picture is that these are now moving in this direction, right? They're going towards opposite ends of the pole of the cell, which makes sense because since the last phase, right, they've been pulled in that direction. Now they're still going. They just happen to have been separated. So if anaphase is dragging them towards the other ends of the cell, then logically, what must be the next phase? What are the chromosomes doing in the next phase? Or where are they, however you want to say it? What's the story for the next phase? No guesses? Actually, before I even talk about that, I'll point this out, even though I'm not going to ask about it. Not only are the chromosomes moving towards the other ends of the opposite ends of the cell, but also the cell itself is kind of going less of a circle and more of an oval, right? It's getting elongated. It's about to split, right? So it needs to get longer, so to speak. Let's see. That'll be the next word for attendance. Polls. I guess, unlike the first word, if you're watching the video later today, not the live stream, but the video that I'm going to record and then post later, so not anybody watching right now, send a picture of a poll. Find a poll and send a picture of that. So again, for the first word, I'm pointing at the word. You need a screenshot of that. For polls, you need to send a picture of a poll. Any questions about this? Make it even easier if you're online watching the video. If you're going to send a picture of a pole, you can send a pole, like a stripper pole or a fireman pole, or somebody Polish or anything from Poland. It's different spelling, but whatever. The world is your oyster. Anyway, anaphase. These chromosomes are being separated, right? They're moving towards the opposite ends of the cell. I was hoping somebody would guess this, but logically, the next step must be they make it to the other end of the cell, right? So this is all very logical. To back up, remember I told you in prophase, they were moving towards the center of the cell. And once they got there, that's what we call metaphase. And then I said in metaphase, they're getting pulled, right? It's a tug of war. And then once they finally separate from that tug of war, that is called anaphase. And then I told you in anaphase, they're moving towards the opposite ends of the cell. Now I'm telling you, once they get there, that's called telophase or telophase, however you want to pronounce it. And that's basically what you need to know about telophase. The cells made it. They're basically the reverse of prophase, which is all very logical, right? So now chromosomes are where they need to be. So we can go ahead and put that nuclear envelope back, right? We can go ahead and unravel the chromosomes. The mitotic spindle can now detach because it's no longer moving the chromosomes around. This should all be very logical. The hardest part, I think, is it's going to be memorizing the names to go along with it, right? It should be very logical, knowing which happens when, but you just have to memorize the names. All right, so as telophase is happening, this is when cytokinesis usually happens. I might not ask about that on the exam because depending on the source, you might get a different answer. And you can look that up for independent work if you want. You could have two different sources. If you can find two different sources, some sources might say it's cytokinesis happens with telophase. Some say it starts with anaphase. Who knows? Maybe there's some other ones. I don't know. But if you find it, let me know for independent work. So anyway, any questions about telophase? Again, mitosis is now done. It's the last stage. They're opposite ends. The mitotic spindle goes away. We don't need to move around anymore. Nuclear envelope forms. Chromosomes start to coil or uncoil. And again, cytokinesis happening. It's starting to happen. Because remember, this whole thing is about splitting the cell, right? Because we started with one cell and we want two. So yes, this is all great because we've split the nucleus. We now have two nuclei, but we haven't split the cell yet. And there is a test question that's going to be about that. And definitely a question on the study guide. Something basically like, if a cell goes through all the stages of mitosis, but not cytokinesis, then what are you left with, basically? And the answer is you have one cell with two nuclei, right? Because if the cell doesn't split, then you now have two nuclei. You have double the genetic material. Two different nuclei. Anyway, any questions about this? All right. There's a picture of it. Here we go. Cytokinesis. Again, your book says it starts with telophase. You might find a different answer depending on your source. So because of that, I'm not going to ask it on the exam. I think it is on the study guide, but I'm not going to ask it. What you just need to know is cytokinesis is when we split the cell itself. It's that simple, right? So mitosis is all about the chromosomes. Cytokinesis is actually about splitting the cell. Your book does point out, though, that it's different between plant and animal cells. And we'll talk about that here in a second. Not that I'm going to ask you. But for independent work, you can look up in other stuff, right? What about, if we're talking about plants versus animals, then what about fungi? What about protists, right? What about the other eukaryotes? Not just plants and animals. Another thing you could really look into is, keep in mind, like I said earlier today and during the last lecture, this is all eukaryotic cells we're talking about, right? This is not prokaryotes. Prokaryotes, like bacteria, they don't have chromosomes like this. They have like one big circular chromosome. Not that you need to know that. But if you want to, you can look that up for independent work too. How do prokaryotes do all this with their one circular chromosome? Anyway, back to this. Let's jump into it. Let's talk about the difference between plant versus animal cells. If you've done those videos about mitochondria and chloroplasts, then you should know a little bit of this already. But let's talk about animal cell first. This whole process of cytokinesis is known as cleavage. I'm not going to ask you that, but your book teaches it, so I brought it up. And I'm also not going to ask you this either. It begins when the appearance of the cleavage furrow and an indentation of the equator of the cell. This wrinkly looking Pac-Man thing, that's a cell, right? And it's starting to split right there down the middle. Honestly, I'm probably not going to ask you anything, right? Probably not going to ask you anything about it. I might, maybe ask you about plant cells. As we get there, you'll see what I'm talking about. But I'm not going to ask about animal cells or cleavage or the cleavage furrow. Plant cells are different, as you should know. Oops, no, there it is again. There's a non-colored version of that picture and a drawing that shows showing what happens. There we go, plant cells. I'm not going to ask you this, but hopefully you know by now that plant cells, unlike animal cells, have cell walls, right? Because we learned about that when we talked about, what was it? The cells, because they don't burst, right? They have the cell wall. Yeah, they have a cell wall. And because of that, their cytokinesis is different. Any of these questions. So just keep in mind, obviously, if it's a wall, unlike an animal cell, things are just going to happen a little bit differently, right? So not only does the cell need to split, the cell membrane, which is what happens in the animal cell, but now we also have to deal with the wall. And yes, technically it doesn't go from like here to here, from here to here. It all starts in the middle. So if you were going to build a wall on the southern part of the United States, between the United States and Mexico, you probably wouldn't want to start in San Diego and make your way down to Texas, right? You probably want to get a bunch of people working all over, right? Got some people working in Arizona, some people working in New Mexico, some people working in California, all building a wall until it all meets up. For the record, I'm not talking pro or anti-wall. This is not that kind of class. I'm not giving you my opinion on the wall. Anyway, any questions about cytokinesis in plant cells? As far as the exam is concerned, again, it's pretty much useless. Just know that because they have cell walls, that the process is a little bit different. All right, now we get into probably the most relevant portion of this. And by relevant, I mean relevant to your life, not relevant to the exam. There's probably not going to be any questions about this. If there is, maybe like one. But let's talk about cancer. Cancer is obviously very relevant. Do any of you know somebody who's had cancer? A lot of people, right? It's there, right? It's a bad thing. Almost lost my mom to it. Anyway, for a plant or animal to grow and maintain its tissues, it has to control the timing of the cell division. So I hinted at this earlier for independent work, right? I said, what is it? It's controlling this. Does it just go on autopilot, or is there something that says, all right, you're in G1, it's time to go to S. All right, you're in S phase, it's time to go to G2. So yes, the answer is yes, there is something controlling it. And I'm about to give you the very, very basic description of it. But keep in mind, it's a lot more complicated, and you can look this up if you want. Long story short, and this is how you need to know it for the exam, the basic answer is there's basically on and off switches, right? There's proteins that turn this on and off, specialized proteins. And it's really complicated. Because first of all, and this is going to be really important later, genes, right? The information encoded in your DNA, that's instructions on how to build proteins, including the proteins that do this. So that's one of the way that mutations can cause cancer. So if you smoke too much, drink too much, or eat too much processed meat, or get too much sun, right? Well, if you damage your DNA, right, you might damage the DNA that has the instructions on how to build one of these proteins. One of the proteins that says stop or go, right? And if you mess that up, then you've messed up your stop and go signals. And then things are on autopilot, and your cells are just constantly duplicating themselves. And that's not good. That's what we call cancer, right? That's basically what cancer is. It's when the cells keep going. They keep duplicating. So again, that's basically it. If you're interested, you can look into it for independent work. Like this is such a rabbit hole. You could have a whole class based on this one slide. You could have a whole biology course based on the information on this slide. all the different things that can affect your on and off switch, your stop and go signals. Because again, they're proteins. If you disrupt your DNA, the instructions on how to build those proteins, then you run the risk of getting cancer. And then there's other things too. Those proteins also interact with the environment. So, there's things like in the water, for example, they're finding, I don't want to get into it, but there's stuff in the environment too that can mess with this, right? So even if your genes are correct and you're making the correct proteins, there are things environmentally that can affect the way those proteins are interacting with your cells, and therefore, again, messing up your on and off switch, so to speak. Anyway, any questions about this side? Cancer sucks. There you go. That'll be the test question. Does cancer suck? Yes or no? Anyway, yes, again, like I said, cancer is a disease of the cell cycle, right? So if a cell has cancer, if it's a cancerous cell, basically on autopilot, they're going and going and going, they're dividing excessively. When that happens, it may invade other tissues. And this is, I've already hinted at this too, but the abnormal behavior of these cells, it begins when the cell undergoes genetic changes in the genes that encode for the proteins in the cell cycle. So I've already told you this. Now you can just see it in writing, right? So there's proteins, again, that control this whole thing, the on and off switches, so to speak. So if you mess with the genes that say how to make those proteins, if you mess those up, then you're going to mess the whole thing up, and that'll give you cancer. Anyway, for the exam, I'm probably not going to ask you any of this. But obviously, this is very relevant information. Probably one of the most relevant things I'm going to teach you in this semester. Because sadly, some of you already raised your hand. You already know people with cancer. Sadly, you're going to, if you haven't yet, you probably will know somebody with cancer. Hopefully, it's not you. Another thing to talk about this for personal relevance, not for the exam. There's a lot of misinformation about cancer. Like, I hate it when people say, like, oh, when we talk about curing cancer, there's all kinds of different cancers, and they're all very different. Because, again, there's all these proteins controlling your on and off switches. So they're all very different. So there is no simple answer on how to cure cancer. We're getting there, though. And we'll actually talk a little bit about it. But, again, are there any questions about this slide? All right. Cancer cells, as I'm sure you know, can form tumors. Tumors are defined as abnormally growing masses of cells. I'm not going to ask you what a tumor is. But again, for your everyday life, that is relevant. Up for independent work, because I'm about to tell you there's something called benign, which is basically it's okay, it's not dangerous. And then there's malignant tumors, which is where they've spread. And obviously, you do need to know that, because I'm just telling you that right now. But another thing you could look up for independent work is look into those benign tumors. What are some of the tumors that really don't hurt anything? On his knee, like knee area, there's a fatty tumor. Completely harmless, actually went away. I have a friend in Seattle who recently just had a tumor removed from her brain, a brain tumor, and yet it wasn't cancerous. So yeah, tumors are interesting, I think. You can look into them for independent work. Really dive into the difference between a cancerous tumor and a non-cancerous tumor. For the exam, I'm not going to go that deep because this is a 100 level non-biology major course. But again, very relevant for your everyday life. Any questions about this slide? All right. Here's a picture. It's a very common type of cancer, breast cancer, right? So there's the breast, there's the glandular tissue, right? The milk ducts, the tumor's there, and then it starts to invade the glandular tissue. And then, your whole body's connected. So then it starts spreading, right? You can see these little dots. It's spread into the lymph system. So it's going to go throughout your body in the lymph system. You can also see that it's in the bloodstream, right? So it can go spread throughout your body via your bloodstream. So again, no questions on the exam about this. This is just an example, but very relevant for your everyday life. And again, another independent work topic is looking into all the different types of cancer. What's the difference between breast cancer and colorectal cancer and and skull cancer. Like she had all kinds of tumors. Worth looking into. Also, how to avoid it. And this is an ever-changing field, right? So I think I heard recently, and I need to look this up. I'll write an independent work paper myself. I think they're out for, colorectal cancer. Now they've lowered the age of when you're supposed to get a colonoscopy because it's becoming so much more prevalent than it used to be. And that's a good independent work topic too. Why? I have hypotheses, but I haven't looked into it. Like it could be our foods, right? Hormones might have something to do with it. So when you eat food with hormones, I won't get into it. We don't have the time, and I'm not going to test you on it. All right. So another thing I'm not going to ask you, but here you go. The three main types of treatment for cancer are surgery, radiation, and chemo. Very obviously, surgery is removing the tumor. Radiation therapy and chemo, as far as we're concerned, are basically the same in that the point is to kill the cells, right? We're trying to kill the cancer cells. Radiation is a little bit more specific, right? You can use it and direct it mostly only towards the cancer cells. Chemotherapy is a little bit less specific, right? You're just basically taking medication that's killing the cells. The bad thing about chemotherapy, I'm sure you've seen people who have no hair. That's why they lose their hair, right? Because not only do those drugs kill the cancer cells and mess up the cell division for the cancer cells, they also kill a lot of your useful cells, and that's why you get sick. Basically, right? I'm making this all very basic. It's simple. It's not all this simple, but for the purposes of this course, that's how we'll describe it. Anyway, any questions about the three main types of treatments? All right. I'm glad your book points this out because there is something else. There's also something called immunotherapy. Back when your book was first published, that was actually upcoming new information. Now it's a little bit more commonplace. As a matter of fact, my uncle has cancer, and they have an immunotherapy because for that particular type of cancer, because it's different depending on what kind of cancer you have. And they said had he come down with that cancer, I think like 6 years ago, for example, he would have been dead for sure. They said it would have killed him. But because of medical technology and the information we've learned, like I said, they have an immunotherapy for that type of cancer, and it saved his life. But anyway, back to what the book is saying. Immunotherapy, basically, again, you're still trying to kill the cancer cells, except now you're using your own immune system to fight the cancer cells, which is interesting. In a really, really broad sense, that's basically like what a vaccine is, right? A vaccine, you use your own immune system to kill a virus, right, or a bacteria, for example. But to do that, you take the vaccine, and that teaches your body how to fight it. Anyway, no questions about this on the exam. Do you have any questions about it? Tumor will be the next attendance word question. If you're watching the video that I'm recording and that hopefully I will post later today, you can take a picture of me pointing at the word tumors. Any questions before I move forward? Cancer prevention and survival. Very, very important information for your life, but not for the exam. I'm sure you already know this. There's a long list, but no smoking, exercise, avoid exposure to the sun, right? Because that'll destroy your DNA, which will make you have mutations on those proteins we talked about earlier. Eat high fiber, low fat. Before self-exams, visit the doctor. That's another good independent work topic because that's a short list, but there's actually a lot of things you can do to reduce your risk of getting cancer. And if you want to, things you can do to avoid getting. Any questions about that? All right. It's a great place to stop. So again, Wednesday is the exam. You don't want to take it on Wednesday or you need more time to take it. You need to let me know immediately. So we won't be back into this until Friday, right? Friday we'll come and we'll go talk about meiosis. Wednesday is the exam. If you're I do post this if you like it. In lab today. Hopefully not, right? No one wants to be there. I'll be there, but I understand if you don't want to be there. And you don't have to be, possibly.